Hey, first of all, good morning, everyone. So my apology for not being able to present this proposal to you in live. Basically, I'm currently attending the National Conference of Clinical Research at the moment. Just to introduce myself, my name is Chian Leong, and I'm currently the professor in cell and molecular biology, and also the Deputy Director of Research at ERDI. Now, the project that I'm going to discuss with you today is basically looking at the identification of metastasis suppressor genes in pancreatic cancer. Now, first of all, pancreatic cancer is considered one of the deadliest cancer, and it is actually very rare. But unfortunately, if you get it, so the survival rate is only about 5%. Now, what is even more important is that the survival rate have not been improved for the past 40 years, despite we are having new targeted therapy and also immunotherapy. One of the reasons for that is basically pancreatic cancer is also highly metastasis, and most of the time the symptom is quite hidden. By the time when the patients discover that they have pancreatic cancer, basically that most of the time the cells have already metastasized to different part of the body. When the cells start to metastasize, the treatment is becoming very limited. Surgery is no longer an option. The only treatment that is available is basically chemotherapy. And unfortunately, for the chemotherapy drugs that is being used, is mainly the gemcitabine-based chemotherapeutic agent, so which has been existed or have been used for the past 40 years. Despite many efforts to actually to improve the efficacy of the gemcitabine, for example, combining it with targeted therapy such as the EGFR inhibitors, you can see from the survival curve here that is actually not much improvement in terms of the survival outcome. So in a nutshell, basically, pancreatic cancer is still one of the most difficult to treat cancer because it is highly metastatic and there is a lack of treatment for that at the moment. Now, when we talk about cancer metastasis, in fact, more than 90% of the cancer deaths are actually attributed to metastasis. If the tumors are actually localized, most of the time it doesn't cause so much of a threat. But unfortunately, when the cancer cells start to migrate or start to invade and metastasize into the secondary organ, what happens is that it will actually colonize the other organs and cause multi-organ failure, and eventually the patient will die from cancer. So if you look at the metastatic cascade, basically it involves a multi-stage. For example, for the primary tumors to actually metastasize, first thing is that there must be enough angiogenesis and also vascularization, meaning that there must be a lot of blood vessels to penetrate into the tumor site. So as the tumor starts to progress and get more and more mutation, basically some of them will eventually be able to detach from detached from one another, then they become encourage independent growth, whereby they can actually intravase into the bloodstream, and inside the bloodstream, then they will then carry all along in the bloodstream and then go to the secondary metastatic site, and eventually they will exit from the bloodstream and then start to colonize the secondary tumors. Now, there is a concept about the metastasis suppressors gene. So just like the tumor suppressor gene concept, the metastasis suppressors are basically a class of genes which normally is when overexpressed, they can actually inhibit the cancer metastasis. So therefore, it is not surprising is that so a lot of the to identify these genes, basically, if we look at the tumor, so it wouldn't be surprised that we will find that a lot of the metastatic tumor cells might have a reduced expression of these metastasis genes. Now, the objective of the study, hence, was actually to identify the putative metastasis suppressor genes in the pancreatic cancer. So we have two approaches to actually do it. The first part of it, we will be using state-of-the-art machine learning or and also large-scale multi-omics data set minings to actually identify the putative metastasis gene. So the first part of the project will mainly involve computational work. And once we have identified the genes, then the second part of the work, we will then perform the laboratory work to actually knock out or knock down the putative metastasis gene and do in vitro cell-based testing to see whether the target genes was indeed a metastasis suppressor gene. So to start with, basically, we will actually use a large size of data set. So from the TCGA, the Cancer Genome Atlas project. Now, the Cancer Genome Atlas project is one of the huge efforts that was conducted since 2006, basically. So the whole project basically profiled altogether the three different types of tumors involving 11,000 patients. And what is more interesting is that in the TCGA project, basically for each tumor, there is actually multiple omics data that is being collected. For example, there is data set for gene expression, DNA methylation, DNA mutation, as well as comprehensive clinical data from the same tumors from the same patients. So that allows us to actually look at the tumors from multi-dimension to actually understand what is the genetic or genomic basis of the individual tumors, for example. Okay, the whole data set basically consists of about, consists of about 20 petabytes of data. Basically, it's a huge amount of data, but in our project, we will be just focusing on one type of tumors, which is basically the pancreatic cancer. So in AIM-1, okay, in the computational work, so we will actually try to identify this putative metastasis suppressor gene, okay, using machine learning of large-scale multi-omics data set using the TCGA data. The first thing we will do is basically just to do a simple statistical association between the pancreatic cancer tumors that are highly metastatic versus those that are not metastatic, and to see whether if there is any known metastasis suppressor genes that is associated with the metastasis status of the pancreatic cancer. Now, the second part of the AIM-1, basically, we will actually use the independent method basically to identify new metastasis suppressor gene. So the method that we are going to use will be using some kind of machine learning techniques. So for example, okay, the KNN or the convoluted network or even the support vector machines. So basically that will allow us to identify independent and new metastasis suppressor genes from the pancreatic cancer data set. So once we identify these putative genes, so one thing we have to be clear is that to be classified and to be actually defined as metastasis suppressor genes, these genes shouldn't affect the proliferation or the survival of the cancer cells. So obviously, if the genes is killing the tumor cells, basically then, well, there will be no metastasis to happen. So again, based on the definition of metastasis suppressor genes, which is actually the genes that suppress metastasis only, but they shouldn't suppress the cell proliferation and survival. So what we are going to do is basically, based on the genes that we have identified, we will then overlay it into a different data set, which is derived from a project, what we call a DRIVE project. The project DRIVE is basically a large-scale RNA interference experiment to knock down each gene at a time to identify which gene is critical for cell proliferation. Basically, by overlapping the two data sets and also our putative metastasis gene, we will then be able to identify the metastasis suppressor genes, which potentially only suppress the metastasis, but not affecting the cell proliferation. So once we get that, then we will actually map it into the whole genomic and also biological pathway to see if the individual genes was actually enriched in certain specific biological pathway or not. So that is for the AIM-1. Now, aim to, okay, once we have identified the genes, as I mentioned, then we will actually do some lab experiment to either knock out or knock down the gene that we are looking for. So once we have knocked it down, so we will look at whether it affects the proliferation use by lab work, basically, and then we will look at whether it affects the cell migration, because one of the hallmark of metastasis is that while the cells will actually will have the high capacity of migrating. So then we will also test whether it can have the capacity to invade across the some kind of what we call the ECM, the matrix. So again, well, if the tumor suppressor genes was, the metastasis gene that is identified is true, when we overexpress them, they should basically stop the migration. And vice versa, if you knock down the metastasis genes, then you should make, then we will anticipate the cells will be much more invasive. And finally, the last part, it depends basically. So we might also want to test whether the cells can, whether the genes that we identify would actually confer or regulate the annoyance resistance. Annoyance is basically a mechanism whereby the cells can actually grow in an attachment independent manner. So as one of the hallmarks of metastasis as well. Now, finally, okay, what is the significance and the implications of this work? I think obviously, if we can identify the candidate metastatic genes, okay, that could actually help us to translate this into a therapeutic target in the future for anti-metastasis therapy. So as I mentioned before, 90% of the cancer cells, the cancer patients' death is mainly because of metastasis. Unfortunately, until today, there is actually no anti-metastasis therapy at the moment. Part of the reason is because we do not actually fully understand how the mechanism of the anti-metastasis, understand fully how metastasis works. So if we can identify this gene, hopefully that will give us some direction for future work to actually translate it into better therapeutics. And finally, this is the team. So that we photo. So we took this sometime just before the COVID in 2019. So as I mentioned, so there are many people here. Some of them have already graduated, but nevertheless, okay, don't worry, okay, if you work in this project, you are not alone. So we have a number of master PhD students, okay, that would actually help to guide you. If not, we have dedicated postdoc fellows as well in the lab to actually guide you on some of the experimental setup. Okay, that's all for now. So if you want to contact me, so this is my contact. So you can either email me, or if you can WhatsApp me, I will be more than happy to answer your questions. So if that's, well, again, really sorry for not being able to be with you today. So I'm happy to take up any questions, or if you WhatsApp me or if you email me. All right, thank you. Bye-bye.